Thank you all for being with us. For those of you who are new or who don't know me, I'm Jim Minow, Executive Director of the CureJM Foundation. Honored to have been a part of this organization for some 8 years now, going into my 9th year. And we've just reached, we've had some tremendous progress over that period of time and prior, actually, as we in 2023 are celebrating our 20th anniversary. Really quite a milestone for a small nonprofit that especially has accomplished as much as this nonprofit has. Particularly honored today to have two giants in research that are joining us and have been through their institutions and individual commitment to the CureJM team. Both of our speakers are focused on research that moves promising therapies into clinical trials. Dr. Joni Rutter is the director of the National Center for Translational Sciences, a center at the National Institutes of Health where CureJM funded a drug discovery program through the Coffey family's generosity that has yielded some promising drug candidates that have been or soon will be in clinical trials for JDM. This has been a very fruitful public-private partnership for CureJM, and we certainly look forward to working with NIH in the next phase of drug development as working with an institution that is as large and extraordinary and committed to developing new and better cures for diseases as NIH is a terrific way for a small organization like ours to leverage our money. Following Dr. Rutter's presentation, we'll hear from Dr. Eric Hoffman. Dr. Hoffman and his colleagues at State University of New York, Binghamton, and ReveraGen Biopharma have in fact been extraordinary partners as we work together to design an FDA-approved clinical trial for the anti-steroid, what I call the anti-steroid vamorolone. Dr. Hoffman will share some of the development story for vamorolone, which we anticipate will be approved by the FDA for Duchenne muscular dystrophy later this month. Our vision is for JM and CureJM to be next up for approval, but we need to accomplish and complete a clinical trial to accomplish that goal. We'll have times for questions at the end of each presentation. So now it's my honor to introduce to you Dr. Joni Rutter, who's the director of the National Center for Advancing Translational Sciences at NIH. She oversees the planning and execution of the center's complex, multifaceted programs that aim to overcome some of the operational barriers that impede the development and delivery of new treatments and health solutions. NCATS focuses on helping to turn promising research discoveries into real-world applications. She received her Ph.D. degree from the Department of Pharmacology at Dartmouth Medical School in Hanover, New Hampshire, and has held various positions throughout NIH and other research organizations in her previous assignments. What you're seeing before you now is a photograph of the NCATS team that we worked with at the National Institutes of Health. Dr. Jim Inglis is on the far left. Next to him is Dr. Travis Kinder, who was the fellow who directed this particular program. And almost all the way to the right, one in from the right, is Dr. Patricia Dranchek, who was also a key part of that team, along with everyone else in the Inglis lab at NCATS. With that, it's my honor and pleasure to turn this over to Dr. Joni Rutter.
Fantastic. Well, thank you so much, Jim. I really appreciate the invitation to be here, and it looks like I have gotten in finally on a different place. So hopefully you can see me. Can you see me?
Yes.
Hey, everybody. Yes. Nice to see you. All right. Okay. So thank you for showing that photo. And Shannon, if you don't mind, can I have you go ahead and run the slides? I don't have them up on this computer that I'm using.
Absolutely.
Yeah.
All right. Thank you so
Thank you,
much.
Shannon.
Well, it's a pleasure to be here, and I really appreciate the invitation. It's fascinating that CureJM is having its 20th anniversary. I will say that I've been at the NIH for 20 years, so we share this anniversary. And I also just want to say how grateful I am to the support of CureJM. I want to thank the parents and the grandparents and all of the patients who have really helped us along the way, not just doing the science that we do, but also learning about JM as well. So with that, hopefully we'll get these slides up shortly. I'm not seeing them just yet, but hopefully they'll be up shortly. All right, yes, there we go. So Jim had asked me to talk a little bit about what we do here at NCATS. And so what I like to say is that NCATS is a place where science goes to become health. And I liken it to this because we do what's called translational science. And so we're moving things from the bench all the way to the bedside. But that bench to bedside, the in-between part is a long and arduous process, and that's the translational science piece, and that's what we tend to do here. So if you go to the next slide, we have a pretty big challenge on our hands. The public health challenge that we tend to face is that we don't study any one particular disease or condition. We study all over 10,000 diseases. So there are over 10,000 diseases, and even with that, there are over 10,000 rare diseases alone. And for those diseases, only 500 of them have a treatment or a cure. So we have 95% of the diseases out there that do not have a treatment or a cure. Not only that, but the time it takes from a particular identification of a potential therapeutic to get that into clinical trials takes about 10 to 15.
Oops. Hold on 1 second. I think we had a little feedback. We'll need to unmute. Sorry, we're having a little bit of a technical difficulties on Zoom. There we go.
Is that better?
Yes.
Okay, perfect. So even with this dire number of 95% of the diseases that don't have a treatment or cure, it also takes a long time for therapies that are being developed to get to clinical trials and even then into market, about 10 to 15 years, and even more so for most of the time. And also with that, it takes about $2.6 billion. So it's a lot of time and a lot of money. And if that weren't enough, there's one more sort of key statistic that we need to understand, and that is about 90% of the time, the promising therapeutic candidates that enter clinical trials tend to fail. They fail generally in the efficacy phase or phase 2 of clinical trials. That's generally about 55% of the time is when they fail in that phase. About 28% of the time they fail in phase 1 or the safety phase. So phase 1 and phase 2 clinical trials are really challenging to get through that particular hurdle to get these promising compounds into people. So how our challenge is really to address all of these numbers. How can we make the science more predictive to get therapies to the clinical environment in a way that we can have more confidence in them when they go in, and hopefully reduce that 90% to a number that's much lower. So these are some of our challenges. And if you go to the next slide, our mission at NCATS is to turn research observations into health solutions by using what we call translational science. And I'll talk a little bit more about that in just a minute. And with this, we are able to develop particular approaches on how we tackle the science that's needed to support these activities, and then we demonstrate their usefulness, and then hopefully disseminate those findings so they can be taken up into the community. And so that's sort of the way in which we tend to think about how we work in the translational science space. And if you go to the next slide, our key goals here are to bring more treatments for all people more quickly. And I mentioned that 5% of diseases don't have therapies. And so we really want to ratchet that number up to about 25%. This is an audacious goal because we know that it's been at 5% for a very long time, and that number is going to be very difficult to move. But we think with the advent of new technologies, new resources, new data science approaches, we have an opportunity to hopefully get to that increased number and maybe even 25%. But I'd rather fail at 25% than succeed at 6%. And so we really want to keep pushing that envelope to hopefully get more treatments to people. So for all people is the middle goal here. This is also a very audacious goal, but we want to dramatically increase inclusivity across every area we support. We want to increase our training opportunities, our postdoctoral opportunities, our career development opportunities, and have more diversity in leadership positions across the research that we support. That diversity of thought, diversity of background really helps us tackle problems in ways that are more innovative. And with that, we hope that we can meet these challenges much more effectively. But in addition to those sorts of things, in terms of workforce development, we want to also make sure that we're addressing health equity and social determinants of health in the problems that we're trying to solve. Because again, these sorts of approaches will get us to the idea of more treatments to people more quickly in a way that's actually going to be workable and generalizable in that. And then lastly, the audacious goal of more quickly. We want to enable diagnostics and therapeutics to reach people twice as fast. We want to reduce that amount of time it takes for things to get into the clinic, and even reduce things like the diagnostic odyssey. We know it takes 5 to 7 years on average for a rare disease patient to be diagnosed. Boy, we'd really like to tackle that particular problem by using a variety of approaches, especially like data science, for example. So these are our 3 audacious goals that we're really trying to move the needle on at NCATS. And if you go to the next slide, I'll tell you some specific ways in which we're trying to get there. So the field of translational science aims to really accelerate the whole idea of translating the research and discoveries by understanding the scientific as well as the operational approaches and principles for how to really get there. And if we can do that, then we can understand those underlying steps in the translational research process that can help make them go faster, better, more effectively, and more efficiently. And so some of the key approaches that we take at NCATS really address that. And I'll just talk a little bit about these here. For example, we want to understand what's similar across diseases to spur multiple treatments at a time. I mentioned that there are over 10,000 diseases. If we go one disease at a time at the pace of 10 to 15 years of getting something into the clinic, that's going to take us thousands of years before we're able to cover them all. So boy, if we can really understand pathway-based approaches to get at more treatments at a time, that's really going to be critical for us. And that's exactly what we're trying to do at NCATS. We want to develop models that better predict a person's reaction to a particular treatment. We know that mice and two-dimensional cells in a dish are not equivalent to a human, so we want to think about how to build better predictive models to be able to make sure that safety and efficacy phase where we're seeing that 90% failure rate, perhaps we can get more confidence in that by using better predictive models. So that's another area where we're trying to build. Next, we want to enhance clinical trials so that they really more accurately reflect the patient population. We want to make sure that we're engaging patients and communities where these diseases are most prevalent in, and bring those into the scientific fold and engage them from the outset, not just when we're ready to do the clinical research, but engage them from the outset to get them interested in the research that we're doing and have them be a part of the value proposition of the science that we support. And then lastly, how do we leverage real-world data and data science approaches to address these public health needs? We're getting much more sophisticated in the data tools that we have available, and we can really be able to bring that power to bear on how we start to move the needle on our audacious goals. And so if you go to the next slide, at NCATS, we study, as I mentioned, all of the diseases out there, but we do have a particular proclivity for rare diseases. And so we are a home for rare diseases. And again, we want to make sure that we're studying those in a more than one disease at a time fashion. So we're trying to develop new ways to understand and treat rare diseases by specifically engaging with patient communities, and CureJM is a fantastic example of this. We want to develop data-driven solutions, and then we want to fund other types of research activities across the ecosystem to help support a variety of work in the rare disease space. And a good example of this is the Rare Disease Clinical Research Network Program, or RDCRN, where they're studying a variety of different diseases at a more than one disease at a time pace. We have 20 different consortia across the country studying different rare diseases, for example, in this program. So these are a variety of ways in which we can support that. Now, if you go to the next slide, a lot of the work that we do is really internal to NCATS. And I say internal meaning that we do this in our NCATS labs, within our labs in Gaithersburg, Maryland. And what we're doing here is to really dig into this preclinical pipeline of drug development. And but our remit is the entire process of the translational science pipeline. It's the preclinical space, but it's also the clinical space too. Internally at NCATS, in our NCATS labs, the bulk of the work is in the preclinical space. And so this is really the idea of moving from developing a target or identifying a target to then doing a large high throughput screening program, then building the medicinal chemistry around any potential chemical compounds that come out of the high throughput screening, and then iterating on those to generate a lead compound to go into investigational new drug status or IND testing. And then we can move that into clinical development. So that's sort of an overall view of this process for the preclinical stages of what we do here at NCATS. And so I'll just say that, again, if you go to the next slide, our whole approach here is not to go necessarily one disease at a time, but we really want to figure out how, using one disease at a time, some of the research projects that we support, like, for example, the work that we're doing in CureJM. These projects, even though they're one disease, are necessary for us to learn from them and apply those learnings to bring them to bear on other areas that we support too. So it's that dual nature of a specific research project that we support, but learning from them, because we know that those projects and what we learn from them can help us break down those barriers that are preventing things from moving very quickly through the translational science pipeline. And so there are bottlenecks, for example, in the areas of database development. A lot of the databases out there are developed. They might be looking at genomics or proteomics or electronic health records, but the data within them are not very interoperable. In other words, you can't really analyze them together because they don't have the same format, and it's very difficult. So it looks almost like gibberish. So one of the things that we're trying to do is to figure out ways in which to make those data more interoperable so that they can talk in an apples to apples kind of a way, and we can integrate those data to get learnings from them. And then, for example, the only other bottleneck I'll point out here again is just that lack of predictive models for preclinical studies. And if we can build platform-based approaches, we can build better human physiologically relevant models that can really inform us as to what might be working very well on the clinical side of things. So these are some of the examples of those little barriers that I mentioned before, those bottlenecks to the translational pipeline. And as we tackle them, we'll be able to move more quickly through that process. And if you go to the next slide, I'll give you an example of some key activities that we're trying to do here in this space. And when we talk about therapeutic development, for example, this process of therapeutic development, again, takes about 10 to 15 years to get through this whole process. And going from candidate selection of a particular compound to lead identification, assay development, pharmacokinetics, pharmacodynamics, and then synthesis and chemistry formulation, looking at toxicology and safety, and then going through all the regulatory phases. All of these types of different chevron diagrams shown here take a lot of time. And so one of the things that we want to do is find ways in which we can reduce that amount of time without cutting corners, but reduce that amount of time. And one of those ways is, if you go to the next slide, is thinking about drug repurposing. And what that does is that chevron diagram that I showed you on that last slide, drug repurposing can essentially take this, cut about half of this time off. That actually could translate into a couple of years as opposed to about 10 years. But essentially, if we screen drug compounds that are already FDA approved, we have a library of FDA approved compounds. So if we screen that library, we might identify a particular compound that is already approved. We already know a lot of information about that. So really, we would just need to focus on the efficacy of that particular compound to understand whether or not it might be useful to put into clinical trials. And this is exactly what we've done for the project that I'll be talking about here very briefly, and I hope will lead into also ReveraGen's proposals as well, or presentation as well. But essentially what we do here is we figure out ways, and you can go to the next slide, we figure out ways of collaborating in the environment here. And so with CureJM, we are connecting with key partners in the CureJM Foundation to engage in the research that we're doing to help us identify those compounds that might be very productive. And you see Travis Kinder here in this photo here, along with others in Jim Glacey's lab. But to provide that scientific and disease expertise and use that as a way in which we can then bring that to the care for the patients, identify ways in which we can then go into clinical trials, working with partners to execute those clinical trials. And there have been a couple of partners that we've worked with for this particular program. And then ultimately, then we come back to the NCATS labs to understand if there needs to be more work done, or maybe where we need to iterate moving forward. But certainly, we started here on this side of the preclinical project for CureJM, where we have taken an assay and a drug screen to identify those repurposed compounds that perhaps might be very useful for CureJM. And so if we go to the next slide, this should, I think this is my penultimate slide. For the high throughput screens that we've done for JM, this is one of the papers that came out a couple of years ago talking about that high throughput screen. But essentially, we identified compounds that inhibit the interferon gamma or interferon beta and MHC class 1 pathway in skeletal muscle for myositis. And then we looked at these drug repurposing screens to find drugs that are already approved for indications that might show efficacy for this new indication, this assay that we typically have performed for juvenile myositis. And we identified 12 compounds that showed promise. Many of these were within the JAK-STAT pathway, and so these are a variety of different inhibitors of that pathway to reduce inflammation. And then there were also some compounds that were identified for glucocorticoids, and then another set of compounds that looked at HDAC inhibitors, which are epigenetic kinds of inhibitors that might reduce interferon beta stimulated MHC1 class expression. And so it indicates that these may be other compounds that could play a key role in the pathways that haven't necessarily been identified before until we conducted this screen. And so we're very much looking forward to understanding how these particular compounds fare as we continue to go through the efficacy parts of the program that we're trying to support here. And I'll, if you click on the next slide, I wanted to just mention and hopefully set the next speaker up go back one, hopefully set the next speaker up very well and from the ReveraGen perspective. But there's another approach on the vamorolone compound that looks to be promising in Duchenne muscular dystrophy, which also has very similar kinds of symptoms related to juvenile myositis in terms of the skeletal muscle pathways, but it may not cover everything. So I think the clinical trials that are done in Duchenne muscular dystrophy will be very informative to understanding perhaps how we might be able to use them for JM as well. And so I look forward to understanding and hearing the next presentation too on where we are in this process. So there's a lot of exciting opportunity here in these compound screens, and I think this holds great promise for what comes next in terms of therapeutic development for JM. So if you go to the next slide, I'll stop there and happy to answer any questions, or we can go into the next presentation and I'll answer questions after that too.
Well, while everyone who has a question comes off mute, you can come off mute and ask it, raise your hand, text it, or put it in the chat. We do have one question for you, Dr. Rutter, by text. You mentioned earlier on innovative new technologies that are moving things forward. Are there any examples or anything you can tell us about any of those technologies? What are some of those new things you're utilizing?
Yeah, so thank you for that question. There are a variety of new technologies, and I'll just say two to give you an example of what I'm talking about. One, for example, is the use of artificial intelligence and machine learning in looking at particular compounds or biological pathways to understand if there might be ways in which we can look at compound development that these may not be approved compounds, but they might help us identify compounds that are not yet approved, but could be very effective in binding to particular proteins or small molecules, for example, in the complex interferon and MHC pathway. And so using that artificial intelligence kind of machine learning tools, that may give us a leg up in identifying more compounds that might be very valuable. The problem with those is, although that's exciting, they'll have to go through more of the thorough process of then developing those molecules as they go through to make sure that they are safe and efficacious through that entire process. So we wouldn't be able to leverage the idea that some of these compounds have already been FDA approved, but it might give us much more powerful compounds that would be useful for JM. The second approach that we're looking into is more gene therapy, gene editing, or antisense oligonucleotide development of different types of modalities of therapies. So we've been talking a little bit more about small molecules here and glucocorticoids and small molecules that affect the signaling pathways. But there may be other approaches that might be useful for other rare diseases that are more about looking at single gene disorders or mutations in those single genes that could be better treated by some sort of gene therapy or gene editing approach. So those are a couple of the ideas that we're thinking about now.
Great. All right, well, we'll pause there. If anyone has, we have probably time for one or two more questions. Jump off mute and ask your question. I know Suzanne, you had one question. Let's see if anyone else has one.
Dr. Rutter, I have a quick question. You had mentioned in part of your presentation that one of your objectives, more sort of downstream objectives in a way, is to enhance clinical trials. And as I think you know, we are very hopeful here at CureJM about 3 clinical trials that we're focusing on. 2 for drugs that were actually identified by Dr. Kinder in his drug repurposing work, kind of building on that, and another drug, one that was repurposed, another drug, deucravacitinib, which was not approved at the time. It was identified by Dr. Kinder. It has since been approved by the FDA, and we're very interested in pursuing an animal model study for deucravacitinib, which would, in a sense, be necessary before we might move into a clinical trial for JM and deucravacitinib. But I guess my question is looking at that clinical trial component, how might NCATS be specifically helpful to CureJM as we look at this next phase of drug development for juvenile dermatomyositis?
Yeah, it's a great question. I mean, one thing is, I mentioned Duchenne muscular dystrophy, and I think learning a little bit more about other diseases that may share common symptoms with JM might be another way in increasing the pool of particular rare diseases that can be studied at once. This is kind of the idea of looking at an umbrella trial where you're bringing in a compound that might be effective for different diseases, but you're able to test them all at once under one protocol. So we're thinking about ways in which to innovate around how to design clinical trials like that, where they're covering a variety of, covering perhaps the same pathway or same symptoms, but may not be exactly the same disease. And this may help us inform what kinds of drugs that are approved might be helpful for them. So that's one thing is really innovating around the design of clinical trials and thinking about that in different ways, and working with the FDA to understand where and how that would be an approach that would be doable.
Great, thank you.
Excellent. All right, well, it looks like we'll go ahead and table the rest of our questions for Dr. Rutter until we hear a little bit about, like she mentioned, corticosteroids and some new options there. So Jim, over to you for introduction.
Yes, I'm now a great pleasure to introduce a friend and colleague, Dr. Eric Hoffman, who is the Associate Dean for Research and Research Development at the School of Pharmacy and Pharmaceutical Sciences, as I mentioned, at State University of New York at Binghamton. Previously, he was an assistant professor of neurology at Harvard Medical School and Children's Hospital of Boston, a professor of molecular genetics at the University of Pittsburgh, and the director of the Center for Genetic Medicine at Children's National Medical Center here in Washington, D.C. He's also the founder of at least two that I know of biotech companies, including ReveraGen Biopharma, which is the company responsible for the creation, development, and ultimate, within the next few weeks, we hope, approval of vamorolone as a steroid alternative for use in this case, Duchenne muscular dystrophy, but obviously we are looking forward to its use in other inflammatory diseases like juvenile dermatomyositis. Dr. Hoffman, over to you.
Thanks. Can you hear me okay?
Yes.
Okay. First, I'd just like to say how delighted I am to share the virtual podium with Dr. Malloy at NCATS. Everything that I show you today would have, as CEO and co-founder, would not have at all been possible with NCATS, without NCATS. Not with NCATS, without NCATS. We were one of the 2011 awardees of an NCATS Trend, Therapeutics for Rare and Neglected Disease partnership, and they brought so many expert personnel within the intramural program to bear on the vamorolone program and really got us off to a strong start. And I think one thing that Dr. Malloy might not have mentioned or as emphasized as much as I would have is how important that was to de-risking the program. She mentioned how many programs fail at different steps. Well, maybe those programs shouldn't have gotten that far to begin with, and so-called late-stage failures. And really that our work with them provided trust in both of ourselves, that we believed in what we were doing because of Trend's input, but also many foundations and other governments. The European Commission ended up funding this for $8 million, multiple divisions at NIH, and 15 different nonprofit foundations also funded this program in a venture philanthropy model. And I can say all of that was due to Dr. Malloy and her colleagues. Okay, so to go on, corticosteroids, they've been around and many people know those. There's 33 members of a drug class used for many dozens of inflammatory disorders. Nobel Prize awarded in 1950 for an impressive effect on about a dozen arthritis patients. Cited by, I heard the Nobel Prize Committee note that this was the fastest Nobel ever awarded. Possibly, I think even faster than the recent COVID one today. But impressive efficacy benefit comes with extensive safety concerns, and that was also noted by the Nobel Committee: bone fragility, stunting of growth in children, mood disturbance, cushingoid appearance, and many others. So what I'll talk to you today about is sort of shown in the right a bit. Blue is prednisone, and what it looks, the crystal structure of it and what it looks like as a molecule. It's pretty small, and it binds to the glucocorticoid receptor where it carries out its anti-inflammatory effects. And in red, we've superimposed vamorolone. And you can see it's very, very similar. There's just this one little kink there where the arrow is. But I'm gonna, in the first couple of slides, get a little sciencey as to what's called structure-activity relationships, or SARs. And just point out two key things that make vamorolone different from all the other corticosteroids in terms of structure-activity relationships, and then go straight to clinical trial data. And as Jim mentioned in the beginning, we expect FDA approval in just a few weeks, and then EMA approval also this year. So the first thing I'd like to point out is the simple tweak here. Can you see my arrow? I can turn it into a laser maybe, but cortisol, prednisone, all these corticosteroid drugs have this key oxygen right at the 11 position. And what's so important in understanding how vamorolone works differently is all corticosteroids have both shuttle between prodrug and drug. So inactive prodrug and active drug. And that's both systemically and at sites of inflammation in the muscle or in bone remodeling or wherever in the body. So it has both systemic and local switching back and forth. And that's done by these two enzymes, HSD11 beta 1 and beta 2. Vamorolone lacks that oxygen. That's the main difference, just that one atom or molecule of oxygen. So we're not a substrate for these enzymes. So we're not modulated by these enzymes either systemically or locally. So we have a differentiation in that all members of the corticosteroid class have an 11 oxygen function on their substrates, where the prodrug is this double bond ketone and the active drug is this OH hydroxyl. But we should not be a substrate for these, and we've seen no evidence of metabolites ever showing up with an oxygen there. And this is unique from all other members of this corticosteroid class. So we've worked with Eric Ortlund at Emory quite a bit and done structure-activity relationships and at the molecular level. And this is from one of his earlier papers, just showing how corticosteroids, this green here, fit inside the receptor. Now here you're lasering in on a part of the glucocorticoid receptor, and here you're now seeing much of the bigger glucocorticoid receptor with the drug in its little green pocket there. And the key thing I want you to point out is this maroon off to the left. These are what are called obligatory cofactors. So basically, once any of these drugs bind the glucocorticoid receptor, they can't do anything unless some co-activators or co-repressors come and partner with them and then carry out the downstream effects. So what I'm going to show you in the next slide, that vamorolone, because it loses this one bond with the glucocorticoid receptor, twists and pushes out that co-activator domain and changes the co-activator domain. That how those proteins bind and which ones. And so here you can see this is actually prednisone in the receptor and vamorolone in the receptor now overlaid in terms of structure. And wherever you see red is different with vamorolone. And again, you see the same area that's red that circles the AHF and the H3 turning red because they're being pushed out into solution by vamorolone. Now what that is then predicts is we're going to see different cofactor binding. And in fact, that's what we see. So these two, NCOR and SMART on the right, are cofactors that mediate the anti-inflammatory effect. And when you see the purple vamorolone go down, that means it's binding much tighter. So it's a much stronger interaction than the other corticosteroids. On the other hand, co-activators, which seem more associated with side effects, are actually binding more weakly with vamorolone than with the other corticosteroids. So now we have two unique structure-activity relationships. One is we're not systemically modulated by these two enzymes because we don't have the oxygen. Second, we're changing how the co-activators and co-repressors bind. So in summary, vamorolone shows two distinct and critical SAR features that are highly and completely differentiated from the corticosteroid class. So the first one is not a substrate for key modulatory enzymes. And again, that's both systemically and at the sites of inflammation. And vamorolone loses a contact site for the GR. And so these strongly predict downstream physiological and clinical differentiation from vamorolone from the corticosteroid class. So now I'll just, last slides go into the clinical trial data. So obviously, I've shown you some chemistry, that turns into theory. But until you prove it in double-blind clinical trials, it won't enter evidence-based medicine. So here's the series of clinical trials we did. The phase 1 trials were entirely funded by foundations, nonprofit foundations under venture philanthropy model. Phase 2 program was funded by NIH and the European Commission, as well as the pivotal study, the double-blind study phase 2. So we did them fairly quickly, even though it was in the time of COVID. And then the last slides, I'll say that just mention 2 trials that are ongoing. So this is the design. I'm only going to speak about the double-blind placebo-controlled trial. It also had prednisone as one of the arms. So it was not only placebo-controlled, but prednisone-controlled. And we did 2 doses of vamorolone, about a 3-fold difference, 2.0 and 6.0. We had a treatment period 1 of 6 months, 24 weeks, and that had all 4 arms. So 2 doses of vamorolone, placebo, and prednisone. And then what's important is the prednisone patients crossed over to vamorolone, as did the placebo patients, whereas the patients that were on vamorolone in period 1 continued on vamorolone. So we had, in a double-blind trial that covered a year, some patients that were treated with vamorolone for the full year, but then half the patients were placebo crossing over to vamorolone or prednisone crossing over to vamorolone. The primary outcome was time to stand from the floor of the highest dose group versus placebo. And then we had a series of what are called hierarchical or sequential secondary outcomes. And that was looking at the other dose and other outcomes like 6-minute walk test. So I'll just show you the 24 weeks and a little data from the 48 week. I should say that this happened in the time of COVID. COVID happened right in the middle. We were very lucky, in part that we were such a small academic focus group. I mean, there's only ever been about 4.5 employees in ReveraGen, very thinly academic type fusion group. We were able to turn very quickly, and we managed to do the time to stand remotely in patients' homes through negotiation with FDA in an acceptable way, using videos with them, working with the parents. And we didn't lose any patients through the trial or any of the primary outcome measures. So it did delay us about 5 months in recruitment because patients couldn't come in, but we finished this trial then in 2021. And this is just the recruitment, 33 sites in 11 countries, went on for about 3 years, and only one assessment was lost, no patients were lost. About 11% of assessments were done remotely. So this is the primary outcome, time to stand. So red is placebo. So you see the deterioration, or stable but slightly deteriorating Duchenne kids that are untreated. And then you see the fairly rapid improvement and stabilization. The high dose seemed to improve more quickly. The low dose still improved. Both were statistically significant. So we hit the primary outcome and the first 4 secondary outcomes, because 6-minute walk test on both dose groups also worked quite well. And time to run walk, the high dose group hit, but just missed on the low dose group. We also had exploratory outcomes of tests that you might be familiar with, North Star Ambulatory Assessment and time to climb, and those were all also highly statistically significant. Now, that was against placebo, but are we as good as prednisone? So here, placebo is the red, you see stable or slight decrease, and then the improvement, and prednisone is green, blue is low dose vamorolone, and purple is high dose vamorolone. And you see the high dose vamorolone pretty much matches prednisone. The low dose, some match, some are a little worse. So less effective, the lower dose in some outcomes. Now, what about safety? So you're fine that you work as a corticosteroid, or are you just another of the 33, number 34 of the corticosteroids? So what is particularly impressive, I know this is just numbers, but bone turnover biomarkers are used in many hundreds of clinical trials to look at whether you're influencing bone catabolism, metabolism, growth versus absorption. And so these are some of the bone biomarkers. And it's very well known, if you look under prednisone, that these all turn very quickly to negative numbers. So all the corticosteroids are known to sort of crash the bone biomarkers. They really just stop bone growth and bone turnover. And with that, in children, you see stunting of growth, where you basically, at least in Duchenne, once you start treating with daily high-dose glucocorticoids, their growth largely halts. So you see these negative numbers all down through the right there. But I just pulled out one in red, but you can see all of them are the same. Both dose groups of vamorolone generally go positive instead of negative. So you hear positive, 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 change from baseline and difference, and these are all very highly statistical significant. So basically, vamorolone does not change the bone biomarkers, as does glucocorticoids and prednisone. Well, is that paralleled with growth? And absolutely. I mean, it's very clear. Kids on vamorolone actually initially grow faster than their peers. It seems to be catch-up growth. They then stabilize after about 6 to 10 months, and then they grow at the same rate as their peers. Now, here's the crossover group you see down in the bottom. Prednisone treated for 6 months, standard slowing of acceleration of growth. Crossover to vamorolone, boom, they just go right back up. So clear differentiation in growth. A key thing is bone breakage. We've measured with Liam Ward in Ottawa, CHIO, spine fractures. It takes a while to accumulate spine fractures, and these are 4 to 7-year-old kids. So all this is preliminary, but in long-term extension studies, it looks very promising. And bone breaks, as far as long bone breaks, will take many years to figure out. So it's a clear win in terms of bone. In terms of BMI, we see a subset of kids in both prednisone and vamorolone gain weight. Doesn't seem to be much of a difference in frequency or degree of weight, and it seems to stabilize after some months. We seem to show less behavior problems. You can see here the prednisone seems to have higher behavior problems than vamorolone or placebo. But you can also see that the numbers are sort of close, and behavior problems are sort of hard to measure, particularly in 4 to 7-year-old boys. Girls, it might be easier. But well, with this, it's promising, but we really can't say anything. So conclusions, vamorolone is safe and effective. Vamorolone's 6 mg per kg is equally effective as prednisone. Vamorolone 6 is safer than prednisone, no stunting of growth, no deleterious changes in bone biomarkers, and possibly less mood disturbances and less steroid-related AEs. And again, I thank all the TRINs, NIH, Department of Defense, European Commission, over 12 international nonprofit foundations. About 5 million was invested by nonprofits, and 20 million will be returned to those foundations over the next few years. And EMA approval is anticipated later this year, and FDA this month. So these were all the sites involved. Now, just 2 last slides I want to show you, 2 ongoing trials. So one is an expanded age range. So this is 2 to 4-year-old DMD boys and 7 to 18. And of these older kids, half of them are transitioning from long-term treatment with high-dose corticosteroids to vamorolone. So we can get some experience in that crossover in practice, not in the context of a clinical trial. This was done in collaboration with Jesse's Journey, now called Defeat Duchenne Canada, and enrolling just in 6 sites in Canada. Enrollment's gone very well. And this is really helping expansion of the label with both EMA and FDA. So we expect a broad age range on the label. And then we've initiated a trial in Becker muscular dystrophy, a milder chronic inflammatory state due to dystrophin abnormalities. That's enrolling in Italy and Padua, and that's funded by the Food and Drug Administration through the Orphan Grants Program. So here, just a slide on these 2 trials, 006, as I mentioned, both steroid and non-steroid, and this Becker muscular dystrophy funded by both NIH and the Food and Drug Administration. One thing with Becker, and this is our last slide, is we have some optimism that Becker might actually respond better than Duchenne to vamorolone. And the reason is we've documented quite clearly in mice and also human muscle biopsies that dystrophin is at a low level in Becker because it's abnormal protein. It's not a fully functional protein, and this seems to be decreased. And what we've shown is it's an inflammation-based RNA, microRNAs, that are binding to the dystrophin messenger RNA and inhibiting it. We've clearly shown those pathways, and that happens in mice as well. It also happens in myositis. So there's some nice publications recently in that in myositis, inflammatory disease of muscle, these microRNAs are induced, they bind the dystrophin messenger RNA and reduce it. And so there's the question of reductions in dystrophin could be some aspect of inflammatory myopathies. So again, as Dr. Malloy, perhaps some more overlap between these disorders. And so here in a mouse model having the same mutation as many Becker muscular dystrophy patients, you can see with placebo, low levels of dystrophin, but it's still there and abnormal. But if you treat with prednisone or vamorolone, those dystrophin increases because you're cutting down on the microRNA, and vamorolone seems to increase more than prednisone. So we're optimistic that this might be benefit added, not only to Becker, but perhaps by extrapolation to myositis. And that's the presentation. I'm happy to take any questions.
Excellent. So everyone come off mute, and we would love to hear your questions for Dr. Hoffman.
Yes, I don't know. Okay, let me just see, where is everybody?
Marge, do you
Marge?
have a question? Is that you, Marge?
Yeah, not so much a question, so much as to thank Dr. Hoffman for all the work he does, because without, people like you so devoted to these diseases, we never would have made as we did. Same with Dr. Rutter. Sometimes it's over my head that I can't get all of it, but I get the gist of it. And I'm very impressed because my granddaughter was from 7 till about, let's see, she's 19 now, till she was about 16. She was on prednisone, and I saw what that did to her. And I should know this because I've asked her before, but she's on one of them that was not approved yet for FDA. I, for some reason, thought it was vamorolone, but I can't guarantee that. But I mean, what a difference. What a difference. She's back to looking like any other 19-year-old. And the other thing I just want to mention, exercise plays such a role in her recovery now. When she was on the prednisone, she really couldn't exercise. But these other drugs, they have so much better quality of life. And I thank you and all your science. Makes a big difference.
Yes. I think you can see, based on Dr. Hoffman's presentation, not only the importance that NIH and NCATS has played in the development of all of these drugs, including vamorolone. Certainly the involvement of vamorolone with NCATS support, was pre-cure JM in that phase. So. But we're happy to see the outcome, and we're happy to hopefully be able to capitalize on some of that outcome as we go forward. Per Marge's comments, I think grandparents and parents, we're all familiar with the side effects and the impacts that prednisone has on JM children, and I think because of that alone, you can see why we are so intent to move this particular drug forward in clinical trials for JM, because if it is approved, or even ultimately in a couple of years, if we're able to use it off-label based upon investigator-led investigations, it has the potential to simply have a huge impact on the quality of life of JM kids. So this is really very much why vamorolone, and as Dr. Rutter said, it's a long journey, from initial studies through translational studies into successful clinical trials. But I feel we're well on our way. The FDA has already responded to our initial clinical trial plan for vamorolone in JDM. We're responding to the FDA's response to bring that, hopefully bring that plan to closure here in the near future. We also have some exciting news, in as much as the rights to vamorolone have recently been purchased by a US-based company, where we're also beginning to develop, I think, some important business relationships in terms of that company's interest in helping push a vamorolone clinical trial in JDM through with the FDA as well. So it's a kind of a very fortuitous circumstance that I think came about for our goals with vamorolone, and looking forward to reporting out to all of you again in the next 2, 3, 4 months as first vamorolone becomes an approved drug, and secondly what that approval will mean for our own clinical trial moving forward.
Excellent. Well, we are going to break into some breakout rooms so that the parents and grandparents and patients on the call can have a chance to interact with each other. So we will thank Dr. Rutter for her very valuable time, and Dr. Hoffman for his as well. Any additional questions that come in, we will make sure that we get those emailed over to you, Dr. Rutter and Dr. Hoffman. But we will let you get back to all of your important work. And as Marge said so eloquently, we are so grateful for all that you are doing.
Thank you. We can't do it without you. We appreciate all of the help that you're providing as well. So thanks to CureJM.
Thank you all.
Thank you.